Well, thank you very, very much indeed. I'm really delighted to be here and see so many of you on this ghastly grey afternoon. And I will be talking about seasonal affective disorder right at the end of this talk, so there might be some traction there. So we came up with this title, Rhythmicity and Coordination, the Importance of Circadian and Seasonal Biology, and I hadn't really quite worked out what I was going to talk about. But before I leap into that, I feel I have to give some sort of explanation, because anybody read the Telegraph here? Perhaps nobody wants to put up there. Oh, they do. Well, I did something in the Telegraph before Christmas, and this is what it turned out like, which is, what do sleep experts do when they can't nod off? And I gave an interview, and it was basically, I listened to In Our Time with Melvin Bragg, as it was in those days. And it always works for me. I'm away in 5 minutes, particularly if it's on sort of Chinese porcelain or something. But anyway, I agreed to do this. I said, "Oh, can we send a photographer around?" And I said, "Yeah, sure, that's fine." So the photographer arrived, and he said, "Oh, we want you in pajamas." And I said, "What?" At this point, my wife said, "This has got nothing to do with me. You're on your own here." So anyway, I agreed and did this rather embarrassing photograph of me looking at my radio in my pajamas. So you can imagine the sorts of response I got from my colleagues. But the best email I got was, "Oh, hello, Professor Foster. I quite enjoyed it wasn't the fulsome praise. I quite enjoyed the article in the Telegraph. But why I'm really writing is that, where did you buy your pajamas?" So anyway, what I thought we would do in the next sort of hour or so, and if you have to leave at 6, please feel free to do so, but I'm here to answer questions until we're finished. I want to start with rhythmicity as a changing concept, and the way that we thought about originally about rhythmicity in physiology originally has changed, and that's sort of the central thesis. Then I want to give, those of you who've heard me speak before, I've tended to do sort of health and wealth type lectures, and there will be a bit of that, but it's largely mechanistic. And I felt that this audience would appreciate mechanisms, because that's largely what I work on. So I want to talk quite a bit about understanding circadian rhythms at a mechanistic level. Sleep regulation, some new and unpublished data, which is currently under review. Then go to a different type of oscillation, which is seasonality, and some recent work regarding how mammals organize their seasonal rhythms, and then finish with humans and the extent to which, and this is sort of a giveaway, we do show rhythmicity, but asking the fundamental question, do we encode day length in the same way that many other mammals do? And that will be the last part of the presentation. So let's think about rhythmicity as a changing concept. And we start, of course, with Claude Bernard. I know that there are young people in the audience. So do they still teach you about Bernard and homeostasis in your A-levels? No? Okay. All right, well, this will be new then. So Claude Bernard was an interesting chap. I think probably not a very nice chap, actually. And he came up, and this is a quote from him: "All the physiological mechanisms, no matter how varied they might be, always have one purpose, that of maintaining the constancy of the internal environment, or the milieu intérieur." And Claude Bernard was a strange man. He did some quite unpleasant experiments, as we would regard them today, and even back then. His wife and daughter came home, and they found him doing some vivisection on the family dog. And at that point, that was the end for Madame Bernard, because she left him. And then she became one of the great advocates of anti-vivisection in France. A strange man. But he pushed the idea of constancy. And this was then sort of developed by Walter Cannon, and we came up with the concept of homeostasis. And what do we mean by homeostasis? Well, this is a classic example of a homeostatic mechanism as originally conceived. So we have a change in body temperature, which is detected by a sensor in the skin and the brain. That information is then passed on to the temperature regulatory centers of the brain, which have a set point, a preferred range in which temperature exists. This then will activate sweat glands. The response is then cooling. And so you have this self-correcting mechanism. And the idea of homeostasis is that you keep everything within a very narrow, constrained range. And you can apply this fundamental mechanism, as illustrated in terms of temperature, to basically any system. A stimulus detected by a sensor going to a control center with a set point, changing an effector, getting a response, and then returning the deviation back to normal. Now, I suppose the point I would like to make is that whilst the concept of a feedback loop has been immensely important in understanding biological mechanisms, and I'll talk about a lot of feedback loops in this presentation, the idea of constancy, I would argue, has been less helpful. And let me explain why. Homeostasis overlooks the importance of rhythmicity and variation in biological systems and regards deviations away from a set point as a breakdown in physiology and not an adaptive response to changed circumstances. And the classic example of this would be, it's striving for constancy. And the classic example would be temperature regulation in camels. This is a wonderful study. So, of course, camels live in hot, dry places during the day, and it gets cold at night. And it can rise to very hot temperatures during the day, and they cool by panting and sweating. But that loses water, and in a desert environment, that can be really dangerous. And so what you see is a change in the set point of temperature regulation depending upon whether the animal is hydrated or whether it's dehydrated. So if we look at the temperature variation in a hydrated animal, we see, yes, it shows more of a variation than humans. Ours is about a degree variation. So it drops to a lowish point at night and then climbs up to a high point during the day. But look what happens if the animal is dehydrated. You get a huge change in the amplitude. There's a change in the set point. The core temperature of the camel can drop to 34 degrees in the early hours of the morning in the cold desert. And then it'll take a lot of energy to rise, to increase the core body temperature to around 40 degrees, and that's the point that the animal needs to pant and sweat and start to cool down. And so by dropping the core body temperature, the set point of the core body temperature overnight, then temperature can plummet, so it takes much longer to get up to those more dangerous temperatures. Classic example of a changing set point, and an example where a breakdown of physiology has not occurred. This is an adaptive response to a particular environmental need. And it's this sort of thing that was, I think, ignored to a large extent in the early ideas about homeostasis. So the point I'm trying to make is that biological rhythms encode environmental and physiological information to regulate and optimize multiple biological systems. And I want to take a look at some of those in a moment. So just a bit of terminology. We see that the general term ultradian rhythms are used for oscillations that are shorter than 24 hours. And so the classic ones here would be an action potential or indeed pulsatile hormone release. And we'll see examples of some of those as the talk proceeds. A circadian rhythm, of course, around about 24 hours, of which there are many. We'll talk about the sleep-wake cycle. We'll see examples of core body temperature changes and hormonal changes such as melatonin. But basically, anything you need to talk about shows some sort of 24-hour variation. Big changes in immune function, for example. And then we'll talk about infradian rhythms longer than 24 hours, such as the menstrual cycle, for example, and I won't have time to talk about that, but that's a really interesting example where circadian rhythms and infradian rhythms interact to generate that reproductive cycle. then seasonal reproduction in mammals, how that's come about. And then I'll finish, as I said, with a seasonal rhythm in humans, which is seasonal affective disorder. Okay, and again, the point I would like to make is that these rhythms don't stand in isolation. We'll see examples where ultradian, circadian, and indeed infradian rhythms all interact to regulate physiology. Now, for the mathematicians in the audience, I thought I would sort of just make the point that despite the huge variation in biological rhythms, they can all be described as physical oscillators using standard terminology that mathematicians and physicists would be very familiar with. So, of course, you have an oscillation, which deviates from a zero or an average, a mean, and you can define specific points on the cycle, which you can call phase points. And the interval of one cycle of phase points we can call period. And of course, it doesn't have to be peak to peak. It can be any easily defined point on the cycle. You can also define these oscillations on the basis of amplitude. And there's two ways of doing that. It's either peak to trough or deviation from the average or the mean. And we'll see examples of this throughout the rest of this talk. And just for one further clarification, frequency, of course, is the number of cycles of oscillation per unit time, per hertz, for example, per second. And so a period is not the same as frequency. Frequency is the number of cycles per second often. Okay, so that was the introduction, and I hope I sort of set the stage. Let's now think about a fundamental understanding of circadian rhythms. And to state the obvious, we sit on an Earth that revolves once every 24 hours. And I think it does need stating because it's so obvious that, of course, the night and the day produce huge changes in the environment. And so at night, it's dark, it's cold. During the day, you can be exposed to UV light, high temperatures, and all the rest of it. And essentially all life on Earth has evolved a response to this profoundly different set of environmental challenges by having a period of physical inactivity, which in us is called sleep. So you're essentially not engaging with the environment, versus a period of physical activity and consciousness where you are very much doing all the things that you need to do, feed, reproduce, and all the rest of it. And of course, sleep and consciousness are always interacting with each other. The state of our sleep determines the state of our consciousness, which is a theme I've developed in other talks, but I won't today. The key point is that the body clock, or the circadian system, anticipates the 24-hour light-dark cycle and adjusts and fine-tunes our physiology in advance of the changing conditions. And of course, biology is highly dynamic. Now, this business of anticipation is critically important, because if our biology simply waited for changed circumstances, let's say waited until it got light to gear up physiology for activity, we'd waste a huge amount of time in that transition where we could be out doing the stuff we need to do. And so the ability to anticipate is one of the really key features of having an internal timer such as a circadian clock. And we'll see again examples of that when we talk about seasonality. Anticipation is key. In these 6 illustrations, the time base is the same, so it's a 24-hour time base from 2 o'clock in the afternoon to 2 o'clock in the afternoon. And the grey bar represents essentially nighttime. And I'll show you an illustration of just one hormone to begin with, which is melatonin. And this illustrates the whole nature of these circadian rhythms, in that what we've got is a rhythm that rises in anticipation of darkness and drops in anticipation of light. And so this is a classic example of anticipation. But this rhythm also encodes something else that we'll come back to at the end of this lecture, which is the duration of melatonin release encodes the duration of darkness. And we'll see that becomes really important. If we look at a range of other rhythms, growth hormone is almost primarily released the first half of the night. Core body temperature We've talked about camels. Ours shows about a degree change, and a subtle drop in core body temperature seems to be quite important for sleep initiation. Cortisol rises in anticipation of activity, mobilizing glucose, increasing metabolic rate ready for activity. Blood pressure drops overnight, then rises very sharply in the early hours of the morning. And in fact, between 6 a.m. and 12 noon is the greatest chance of having a stroke, partly because of that sharp rise in in blood pressure, delivering oxygen and nutrients in preparation for activity. And our ability to process information shows a huge variation across the 24-hour day. In the early hours of the morning, our ability to process information is worse than if we'd consumed enough alcohol to make us legally drunk. It's that level of impairment, which accounts for the fact that if you're driving a car in the early hours of the morning, then you're at much greater risk of having an accident. 200,000 crashes on the American motorway system are associated with loss of vigilance and falling asleep as a result of microsleeps in the early hours of the morning. Okay, so you get that extraordinary tuning, change of our biology to cope with the varied demands of a 24-hour day. And I suppose, again, a key point is that the circadian system delivers the right stuff in the correct concentration to the right tissues and organs at the correct time of day to optimize our biology in a 24-hour world. It's extremely fascinating and dynamic. And the key point is that without the time structure provided by the circadian system, our physiology, including our sleep-wake cycle, is just plunged into chaos. And I just want to, this is the only bit of the health and wealth lecture that I wanted to go into, because I really want to illustrate the impact of a breakdown of that time structure, a breakdown of the circadian system, and the consequences of sleep and circadian rhythm disruption to our biology and our cognition. So you can conceptualize sleep and circadian rhythm disruption in this sort of schematic here, where the most obvious is shift work, but extended hours and our 24/7 society, neurodiversity, where scarred sleep and circadian rhythm disruption can be very marked, light and noise pollution if you live by a station or a road, overall levels of stress and illness can drive scarred. And it does two broad things. There's physiological disruption, and then there's psychosocial disruption. Let me just unpack that a little bit. So physiological disruption, so those hormones we talked about, that beautiful sort of set of coordinated responses, become uncoordinated and they start flying all over the place. So reproductive hormones, growth hormone, the metabolic hormones, leptin and ghrelin, all start falling out of alignment with the demands of a 24-hour rest activity cycle. We see toxin clearance, the clearance of things like beta amyloid from the brain can be massively disrupted with sleep and circadian rhythm disruption, and indeed, of course, memory and cognition. This cycle interacts with psychosocial disruption, which is, you're impaired and you have difficulty coping. That can lead to things like low self-esteem, social issues, with partners, friends, colleagues, frustration, anger, increased worry, anxiety and depression. And essentially what you get is emotional, cognitive and physiological pathologies as a result of that mistiming and disruption. And of course, they can promote more scarred. And so you have the first example of a feedback loop. Okay, let's look at some of the consequences of scarred. And as I say, I'm not going to go into any great detail here, but I just want to sort of emphasize what happens when you take away that timing signal. So in terms of our emotional responses, you get fluctuations in mood, irritability, anxiety. Loss of empathy is a really interesting one, where you fail to pick up the social cues from friends, family, work colleagues. Risk-taking impulsivity. If you're driving, I'm sure I can get through that traffic light before it turns red. No, you wouldn't. Why would you ever think of doing it? But that's what happens if you have scarred. Negative salience, very interesting data there. The tired brain and the disrupted brain remembers negative experiences but forgets the positive ones. Increased use of sedatives, alcohol, illegal drug use. Cognitive impairments, well, everything starts to fall apart. Your ability to multitask, your memory consolidation, concentration, communication skills, decision-making, creativity, productivity, you name it, it starts to fall apart. And then long-term, as you see in long-term night shift work, you have constant chronic daytime sleepiness and microsleeps, but cardiovascular disease, we'll talk about some of these parameters when I talk about the profoundly blind fairly shortly. Altered stress responses, which lowered and therefore lowering immunity, which may be the basis for high rates of cancer. The rates of cancer in night shift workers are now so well documented that the World Health Organization has classified night shift work as a probable carcinogen. Metabolic abnormalities, type 2 diabetes, mental illness, and dementia. So the key thing is that SCARD, circadian rhythm, sleep disruption, is not simply feeling tired at an inconvenient time. It has a global impact upon our health. So biological rhythms encode environmental and physiological information to regulate and optimize multiple biological systems. And here we see examples of where this system has broken down. Oh, yes. So this is the book I wrote a few years ago. If you are interested in the breakdown of circadian sleep systems, then you might want to take a look at this. Okay, so let's now move to mechanisms and the generation and the regulation of circadian rhythms. So if we look to the anterior hypothalamus, so there's the optic nerve, and just above the optic nerve is a tiny structure in the hypothalamus called the suprachiasmatic nuclei. So this is the optic chiasm where the optic nerves fuse, and just sitting above this, in humans is a paired structure. That's why it's nuclei, not nucleus. Nasty pedant there, but anyway, it's about 50,000 neurons either side of the brain. And one of the, I think, incredible triumphs of neuroscience over the past few decades has been an understanding of the nature of this master pacemaker within the brain. We used to think that 24-hour oscillations were simply a product of cell-cell interactions. And in the early days, we used to hang out with people working on locomotor rhythms because we thought we could learn about the circuitry. But we now know that each cell has the capacity to generate a circadian rhythm. And there are a bunch of clock genes. I'll go into more detail for of these mechanisms in a moment, but this schematic will suffice. So clock genes undergo transcription and translation to produce clock proteins. The clock proteins then move into the nucleus and inhibit their own genes. So there's another classic example of a feedback loop. Then the proteins are degraded, and so the clock genes can once more produce clock proteins, and you have this wonderful negative feedback loop. And that's at the core of the ability of a single cell to generate a 24-hour oscillation. And so here we see a cartoon of that oscillation. Now, that's the driver that is coordinating the rhythmic activity I talked about earlier, getting the right stuff to the right tissues and organs at the right time of day. But it's no use if it ticks away in isolation from the external world. And for almost all circadian rhythms, the key factor regulating, adjusting the internal day to the external world is exposure to the light-dark cycle. And that is detected by photoreceptors within the eye, and I'll come back to those fairly shortly. Okay, now that molecular oscillation is then translated into a physiological response. And so here we see a molecular rhythm, and this is an example of how that molecular rhythm changes the physiology of the master clock, so that we see that during the nighttime, the firing is asynchronous, but during the daytime, it's synchronous. And this different pattern of electrical activity is sort of partly encoding our ability to sleep and wake. And I'll come back to the mechanisms underpinning that fairly shortly. Okay. As I said, we thought we understood the circadian system. First of all, it was a network, and then we realized it was a whole bunch of cells in the SCN driving rhythmicity, but it actually wasn't driving rhythmicity. What we didn't appreciate is that every cell in the body has the capacity to generate a 24-hour oscillation. And so, again, a change in the way we think about circadian rhythms. It's not that the SCN is driving 24-hour rhythmicity of the sort I showed you earlier, but it's actually coordinating billions and billions of individual oscillators that are scattered around the organ systems of the body. So what we've got is not a clock, and that's what we often use as a shorthand, the body clock. It's actually a circadian system, an extraordinary network of biological time, doing the right stuff at the right time. Now, I want to come back to the importance of entrainment. And so I said that the light-dark cycle is the key factor in making sure that the internal phase of the biological day is locked onto the external world. But if you are under constant dim light, or if you've lost your eyes, and I'll come back to that later on, then what happens is the clock continues to tick, but it drifts out of sync with the outside world. And so the big question that I got interested in a few years ago is really, how is the master clock, the SCN, entrained by light? And so we looked again at the biology of the eye. And this is something that I suspect most people will be familiar with. You have the retina illustrated here with the rods and the cones, and then the inner retina doing the first bit of visual processing, and then the ganglion cells, which is sort of trying to make some sense of that light-dark information, firing an action potential, which is the first time you get action potentials in the retina, and then this goes off down the optic nerve into the brain, where it's turned into a representation of the world. The problem I had was that what the visual system does is grab light and then in a fraction of a second forgets it's seen it to try and make an image of the world. And what the clock needs is almost the exact opposite. It needs to get an approximal, an indication of the amount of light over an extended period of time. And I suppose, as a sensory biology question, I couldn't understand how the classical visual system could also gather light information over extended periods of time. And when I mean extended periods of time, I mean, we're talking in terms of the visual responses in fractions of a second. But for the circadian system to work, it has to integrate information over minutes and often hours, and it's bright light. So these are very different sensory tasks. And so how does the eye do it? And we looked at the retina again, and we started working on mice with hereditary retinal disorders, mice that were actually being studied to understand human visual defects. And I'll cut 15 years of my life short by just summarizing the fact that we eventually got to a mouse model where all the rods and all the cones, all the visual cells had gone as a result of either natural mutations or genetic ablation. And when we put these mice in a running wheel and exposed them to a light-dark cycle, they would show, of course, oscillations. But the thing that blew our socks off is that these oscillations were completely and absolutely coordinated with the external light-dark cycle. So we discovered fairly early on that you could be visually blind, but not clock blind. So the obvious question is, well, is it in the eye? Well, when you cover the eyes, this response is gone. So something else had to exist within the eye. And I was talking to Anne Grocock earlier, who reminded me that when we first proposed that there might be another photoreceptor in the eye, different from the rods and cones, my vision colleagues were completely incandescent with rage. One person, and I sort of said, so the data are consistent with another photoreceptor in the eye. One person stood up and looked at me, and I thought, oh, they're going to ask a question. They just looked at me and said, bullshit, and walked out. And then another person said, shouted, are you seriously considering that we've been studying the eye for 150 years and all of us have missed another photoreceptor in the eye and you found it? And of course, I was much younger in those days and full of sort of confidence. I said, yes. But actually, it took 10 years to convince the vision community, and then further time to show that, well, what is that photoreceptor? We were able to identify it in mice and David Burson in rats, and it turned out that these ganglion cells, 1 out of every 100 or so, is directly light sensitive using its own photopigment, its own light sensitive molecule, which peaks in the blue part of the spectrum. So there's a third receptor system within the eye. Well, how does light regulate the molecular clock work? So I want to go back to that feedback loop I talked about earlier, this negative feedback loop of the molecular clock. And so let me just briefly outline what's going on. So if we take one of those SCN neurons and we take the front of the clock off and we look into the nucleus, you'll see 2 classes of genes. There's the PER genes, 1 and 2, and the CRY genes, 1 and 2. And they have a key regulatory element called an E-box. That E-box is bound by these regulatory elements, clock and BMAL, BMAL1, and they activate transcription and then translation. So you get these proteins and these protein complexes forming in the cytoplasm, and it's more than just PER and CRY. Other proteins are involved as well. They move into the nucleus, inhibit the transcriptional drive by clock and BMAL, so you lose that drive, and then the proteins in the cytoplasm are degraded, then in the nucleus, and you start again. So that's the fundamental negative feedback loop that underpins those circadian rhythms we talked about. So you have a negative feedback loop and a beautiful oscillation. Okay, that's great. But where we came in is we want to understand how that molecular oscillation is aligned to the outside world. We discovered a new set of photoreceptors, but what are they doing? How are they interacting with this feedback loop? And there are 2 regulatory elements on PER1 and PER2 called AP1 and CRY. And they're well-known regulatory elements. And so that's what I want to focus on next and explain how light changes the pattern that allows entrainment. So light signaling to the molecular clockwork. And I would like to acknowledge my 2 very close colleagues who I've worked with on this, Arti and Shri. Absolute delight to work with, and just brilliant individuals. So the work I'm going to be talking about is very much the product of our collective work. So, okay, here we have our PER1 and PER2 genes with the regulatory element, the AP1 and CRY. So I'm now going to activate this sort of cartoon. Here's our photosensitive retinal ganglion cell, and it will detect light. It will then send action potentials down its axons, and depending on the frequency of those action potentials, will then release 2 neurotransmitters, glutamate and PACAP. They, in a series of steps, will increase intracellular cyclic AMP and calcium, these important signaling molecules. Those 2 acting together then start to activate 2 further regulatory elements. So cyclic A activates protein kinase A, which then phosphorylates CREB, which is that little star thing. Calcium in particular activates calcineurin, which then dephosphorylates CRTC1. And this is a really important step. It's taking away a phosphate group from CRTC1. And under those circumstances, it can then bind to phosphorylated CREB. That complex can then bind to CRY, and you get the first stage of the transcriptional drive of those PERs. So PER1 and PER2 transcription, and the first wave of the production of the proteins. But there's a second mechanism. FOS, which is an immediate early gene, also has a CRY element, So it will be activated and phosphoprotein is produced, but it needs a binding partner. And that is in the form of phosphorylated Jun. And Jun is phosphorylated by ERK. So you get this second wave of PER1, PER2 transcription and translation. Okay, so those increased levels of PER1 and PER2, depending upon where they occur in the 24-hour cycle, will either advance or delay the clock and set it to the external light-dark cycle. The thing that interested us as well is that, okay, so this is the system that turns it on, but when you look at the pattern of gene expression and protein expression of the PER1 and PER2, they go up. They go up at slightly different levels, and that's because of the change in the numbers of AP1 and Cre binding domains, but they go down. And nobody really asked the question, well, what the hell turns these things off? They're only useful if they are on for a certain transitory period. So we then discovered that light also induces another gene and protein called SIC1. Again, it's that Cre element. So that Cre element will be activated by Krebs, CRTC1, produces SIC, and it's translated into a protein. And this is the bit I asked you to remember, because this kinase then adds a phosphate group to CRTC1. And of course, in the phosphorylated state, it can no longer bind to Krebs. So the whole transcriptional drive then collapses. So there's a feedback loop whereby the effects of light are limited because of the activity of this kinase. So you get the breakdown of the complex, you get loss of transcriptional drive, Cre is also, SIC1 is also turned off. Then if we turn the lights off, the whole cascade falls apart. So what was exciting is that we had a pretty good idea at a molecular level how light was interacting with the molecular clockwork. And what I found exciting is, well, how light also turns it off. And so I want to just sort of talk about that negative feedback loop of SIC1, because I think it's so cool. And I apologize for, please indulge me. So this is what we did. We put mice in running wheels. And you see that the grey bar is when it was running in a running wheel. It's a nocturnal animal. And then what we did is that we advanced the light-dark cycle by 6 hours. And we just sort of measured how long it took for the normal mouse to adapt to the shifted light-dark cycle. Then we took a mouse in which SIC1 had been turned off in the master clock. And we did the same experiment. So here's normal entrainment to the light-dark cycle, advance the light-dark cycle. But what was so exciting is that instead of taking 6 days to adjust, it adjusted very rapidly within a day. So what we discovered is that SIC1, and that's a cartoon. These are the real data. So this is the real running wheel activity. You can see it drifting. And then the SIC1 mutant, you see that it adapts, it aligns beautifully. Now, I suspect some of you are already onto this, because yes, SIC1 acts as a brake on the effects of light on the clock. What are the implications of this? Well, yeah, SIC1 is why you get jet lag. So the next time you're staggering through the airport and your clock has failed to adjust, it's because of SIC1. What I'd really like to do, because there's lots of people that come up to me and say, oh, yeah, I never get jet lag. And I would like to sequence their SIC1 gene and just see if there are any polymorphisms in it, which might account for why they generally might not get jet lag. Okay. So I said I wanted to talk about profound blindness. And so, yes, you can be visually blind, but as long as you've got your eyes, you don't need your rods and cones because you've got that third receptor. But what happens if you've lost your eyes completely? And I've been privileged to work with Blind Veterans UK. And so this is a study where we measured melatonin. And melatonin is tightly regulated by the clock, as I showed earlier. It rises and it peaks at around about 4 o'clock in the morning. And this is the melatonin pattern over a 4-week period of a person with normal eyes. And you see that the peak in melatonin occurs at the same time every day, beautifully entrained. In this poor individual, they lost their eyes as a result of combat. And you see that the peak in melatonin, well, it's drifting through time. It's getting later and later and later. When you plot that, you see that it's completely out of sync. These individuals are time blind. And I think it's really important to appreciate the impact of that. So these are quotes from some of the people I've worked with. It feels like constant jet lag. I feel ill all the time. I'm at my wit's end, suffering from variable bedtimes and wake times. I'm slowly becoming socially isolated because there are periods when they're aligned to their family and friends, and then they'll drift off again. It's really awful. And I could tell you lots of examples. I will just mention one more, where one chap told me, he said, look, I can kind of adapt to the fact that I can't see. What I can't adapt to is my body constantly fooling me about the time of day. So this is a really serious issue. The eye is the organ of both space, vision, and time. And in some sense, losing one's sense of time seems to be more profound. This is the health status. I mean, I said what happens to circadian rhythm disruption. This is the study group that we worked with. We looked at the percentage of individuals with various conditions. You see that almost 70% have cardiovascular disease, mental health conditions, autoimmune. I mean, it's just the most appalling health status. Now, that's not going to be completely due to circadian rhythm disruption, but that will form a major part of this appalling health status. Biological rhythms encode environmental and physiological information to regulate and optimize multiple biological systems. And we see here a complete breakdown of that system. So what can we do? Well, we can start to translate some of the fundamental information that we've been working on over the past few years about how the molecular clockwork is regulated. So here's our schematic again. And what we've been able to do is, and I won't bore you with all the details, is identify compounds that will mimic the effects of light on the clock by upregulating cyclic AMP and calcium. So what we then went on to do, and we did this first in cells, then in mice, and a range of different techniques. But I just want to show you what the latest is. So this shows a mouse running in a running wheel in complete darkness. Its body clock is slightly shorter than 24 hours, so it'll get up a little bit later and later and later. A bit earlier and earlier and earlier each day. And then we gave the drug, and to our intense excitement, the drug acted just like light. The activity locked on. When we took the drug away, it drifted off. So that's the cartoon. Let me now show you the data. This is no drug, and you see the rhythm drifting through time. And again, this is injecting the drug on a daily basis, and the clock locks on, take the drug away, and it drifts off. So we have, really for the first time, a biological mimic for the effects of light on the circadian system. Where we are, where we are is we've done all the mouse work. We've actually looked at the effect of the drug in humans, and there are no safety aspects. And what's exciting is that it's only effective for 2 hours, which is important because that's very much like light. It's a short hit on the clock. Okay, so we're now getting ready with our friends in Australia to do a proof of concept, which is very much like the mouse study, which will be to keep humans under constant dim light, give them a daily injection, and show that the clock will lock on. And I think the exciting thing for me is that I genuinely feel that we can begin to think about this drug as a way of giving back a sense of biological time to the time blind. So exciting times. And I think actually it's a good example of where fundamental curiosity-driven research is being translated into a really tangible clinical result. Okay, sleep regulation. Don't worry, not all the topics are as long. How are we doing? Oh my God, look at the time. Sleep regulation, it's more than just the clock. We started late anyway, didn't we? So very briefly, So sleep regulation is essentially, you can think of wake being the activation of a whole bunch of excitatory neurotransmitters bathing the cortex during the wake state. And then that's opposed by sleep promoting systems, which are turning off those systems. But also in the midbrain, there's a wonderful interaction that generates this REM, non-REM cycle. And actually during REM sleep, acetylcholine is actually turned on again. But you also have muscular paralysis during REM sleep. Sometimes that muscular paralysis doesn't occur, and there's this condition called REM behavioral disorder. And those individuals don't show paralysis while they're dreaming and act out their dreams. And there have been several well-documented cases where husbands have killed their wives thinking they're an intruder and haven't been prosecuted because they had no knowledge of what they were going to do. Yes. But the sleep-wake cycle is regulated by two processes. The circadian drive, which is what I want to talk about now, but also sleep need. But let me just sort of explain how those two systems interact. So when we're active, the sleep pressure, the drive for sleep builds. And we and others have shown that that's the buildup of a substance called adenosine within the brain. So when we're active, it rises, and when we sleep, it dissipates. But of course now, your adenosine levels will be, it's late afternoon, will be quite high. So why aren't you falling asleep? I can't see anybody asleep, so that's a big relief. And that's because of the drive from the circadian. Oh yeah, the other thing I should say is coffee. The reason coffee works is that it blocks the adenosine receptors within the brain. That's why you actually feel more awake, because you're blocking the sleepiness signal to the brain. So that sleepiness drive is opposed by the circadian drive for wakefulness. And so it's low during the first half of the day, but it builds, keeping pace with the drive, the sleepiness drive, and ideally exceeding the sleepiness drive. And then towards bedtime, the wakefulness drive drops, you sleep, and during sleep, the circadian drive for wakefulness is low until the next morning when you wake up. And then, of course, during sleep, you have that cycle of REM, non-REM activation. And I think in view of the time, I'll just skip that slide. The key thing we've been addressing recently, and this is some unpublished data, is, okay, what's that signal from the SCN? How does it work? We know mice are nocturnal, of course, and during wake, there's this low frequency, incoherent firing activity. And then when they're asleep, it's a beautiful, coherent, rhythmic system. And so how does the ultradian pattern emerge? How does this ultradian pattern get influenced by a circadian pattern? And in brief, so in the wild-type mouse, we have a night signal that goes to a bunch of receptors that promote the release of calcium. That activates protein kinase C gamma, which is a very promiscuous kinase that then interacts with multiple different pathways and leads initially to the overactivation of multiple pathways linked to brain arousal. So you've got yet another amplification. There's just masses of stuff going on. And that seems to lead to this incoherent firing pattern in the master clock, that the default state is wake. But what we've been able to show is that during the day, augmented by light, there's a much greater level of calcium release. That promotes the release of SIC1. We've already seen SIC1 is involved in jet lag. Now it's acting as a kinase to inhibit protein kinase C gamma. So what happens is that it essentially takes away that massive over-amplification system, calms everything down, and allows a coordinated output from the SCN to give the appropriate signal for the sleep state. So you have a quenching of the arousal pathways within the brain. Now, I'm not going to show you all the data for that, but I'll just show you one really cool movie, which is related to the physiological evidence. And what I want to show here. So this is some wonderful work done by Marco. And so, as I say, the synchronous firing pattern leads to sleep. And so here's the SCN using reportadine, and you just obviously speed it up over 24 hours. You see that beautiful rhythmic output. And then if you take away SIC1, which is acting as that quenching agent for the protein kinase C gamma, you see a beautiful asynchronous. Look, it's all over the place, and that's what is leading to the wake state. So we just submitted. We heard today that the paper's gone out for review, and so I think this is really exciting times. We're getting some really fundamental understanding of what's going on and how the circadian system is regulating the sleep-wake cycle. Seasonal world, don't worry. I know we're overrunning, and if you have to leave, I'm going to be another 10 minutes. I apologize. So I want to talk about seasonal rhythms. And of course, this hardly needs an explanation. The Earth is tilted on its axis as it goes around the sun. So on the 21st of June, when the northern hemisphere is pointing towards the sun, it's our summer. And of course, in the south, it's the winter. And so you've got this essentially wonderful change of day length across the year. Now, this is an example of some of the day length changes that you'd see at 2 latitudes. Somewhere at our latitude at 50 degrees north, you get this over 16 hours of light during the summer, and then mid-December, less than 8 hours of light. And at 5 degrees north, that amplitude is much squashed. Okay, so the key thing is that huge amount of light energy is driving plant primary productivity. So if we look at this map of the Earth in June in the northern hemisphere, that red or yellow is productivity, it's plant growth. And if you look at the same pattern during December, it's essentially very little plant growth, if anything. Now, the key thing is that the availability of food around the time of birth is the vital factor for determining when young should be born. And getting the timing wrong means death. So let's think about how you get these seasonal responses in mammals. Because young can't be produced instantly, if you used food, lots of food, great, I can start reproducing. The time you actually produce the young, the food, the good times will have been gone. So you again need a mechanism to anticipate the good times. And this proximate factor is day length or photoperiod. And let me just give you one example of this. And here's a small mammal, something that you might be familiar with, a hamster. It takes 30 to 60 days to get the reproductive system up and firing live rounds, and then about 15 to 30 days gestation in the uterus. It takes 2 to 3 months to produce offspring. So if we look at some of the events here. Here's the changing day length. And here's the sort of 2 to 3 month window where stuff needs to happen. So what happens is that the reproductive system is triggered with the increasing day lengths of spring, early spring. So the testes, for example, show massive changes. The paired testicular mass of a hamster in full breeding, the combined weight of the testes is greater than the weight of the brain, which is extraordinary. Then you can produce active sperm, and this can lead to the production of offspring, and they're produced at a time when there's lots of food, so the mother can wean on milk, and then can feed milk, and then the youngsters can then feed off of the plant productivity. There's time for growth before hibernation in the winter, and then of course the whole cycle starts again. So one of the extraordinary things over the past few years is an understanding of how that process takes place. How is day length encoded biologically? And I just want to give you a sense. I'm coming back to pineal melatonin, because what I kind of said earlier is you've got this light cycle detected by the dawn-dusk cycle, duration of darkness detected by the SCN, and via a multisynaptic input to the pineal, you've got the production of melatonin. And the key point is that the duration of dark determines the duration of melatonin release. So you've got a biological marker of the length of darkness. And of course, during the spring, the night length is decreased, the pattern of melatonin is decreased, and it's these signals that are used to regulate reproduction in all mammals, or rather all seasonally reproductive mammals. And again, just to orientate you before I jump into the pathways, we're looking at the hypothalamus, and the hypothalamus has these gonadotrophin releasing hormones, which pump their neurohormones into the blood supply that feeds the pituitary gland. This then, depending upon whether the GnRH neurons are stimulated, will change the pulse of the gonadotrophin release into the vessels and the pulses of the reproductive hormones. And ultimately, that change in pulse frequency will change the reproductive status of the males and the females. Now, in the cartoon that I'm going to show you, there are two bits of the pituitary I want to draw your attention to. There's the pars distalis, the pars tuberalis. They're different bits of the pituitary gland, but they play very important yet different roles. Okay, so the first thing is that the pars distalis of the pituitary gland normally produces thyroid stimulating hormone. That's released into the blood, and that stimulates the thyroid gland to release into the circulation the inactive form of thyroxine, T4. And that goes around into the blood, and as we'll see, it eventually gets to the hypothalamus. Now, the encoding of day length by melatonin goes to another bit of the pituitary gland, the pars tuberalis. And short nights, long days, stimulate the pars tuberalis again to produce TSH, but it goes to a different place. It's not regulating the thyroid, it's going to the hypothalamus to produce an enzyme that converts T4, the inactive form of thyroxine, into T3. T3 then stimulates the gonadotrophin releasing hormone cells that then go to the LH, that release LH and FSH from the pituitary gland, and then go to the reproductive organs. And so you've got this incredible, beautiful mechanism for the regulation of seasonal biology. So, final bit, and it will only be at 5 minutes. What about us? Have we or do we show seasonal rhythms? And the assumption has always been, no, of course we don't. And we certainly don't show anything like a hamster. Our testes don't become the weight of the brain. I think that's kind of obvious. Well, not for most of us anyway. But these are some studies done by a mate of mine, Till Renneberg, based in Munich. And he looked at birth records from Spain from 1900. And you see that there was a peak in conceptions until about 1940 of about 30%. There was a huge rhythm in, and he actually got birth records and then counted back to get conception records. Then the end of the amplitude decreased, and then you had World War II, and then Franco and industrialization in Spain, and you get a drop in the amplitude of these oscillations. They're barely detectable, but in many societies there's a very, very low amplitude rhythm. So what's the driver? Is it photoperiod and melatonin, as we saw in the hamster? No. There's no evidence, direct evidence that we have that changing day length, changing the pattern of melatonin is underpinning that change in births historically. Seasonal nutritional health. Now, this I think is a probably more likely cause. Before the Industrial Revolution, there was a peak to trough rhythm in births of about 60%. It was huge. And before industrialization, of course, there was a huge variation, and conception occurred in June, July, August. And the argument here is that people were living on the margins of, they were not well fed. And there were periods of the year when they were pretty low on calories. But at the end of the spring, summer, there would be lots of food around. And it was the food status of the mother that would determine conception. And there's some pretty good evidence that might indeed be the case. So, for example, we do know that in women, low nutritional status means that they will not have periods, they'll have amenorrhea. And so there's some very interesting thoughts about that. Seasonal environmental temperatures, well, there are some interesting evidence from the southern United States that there's a really conception tends to be during the winter. And the argument here is that during the hot southern states, high temperatures decrease sperm production. So there's some reasonable data suggesting that testicular temperature, there's a 14% drop in sperm production. And is that correct? We have an expert here who can correct me if I'm wrong. Jump in. That's correct. Great. So maybe part of it is temperature of the testis. See a societal influence. This is a great one. So when the Inuit were studied in this group here, it was shown that there was this peak of births in the beginning of the year. So births occurred during the first half of the year, and there was lots of excitement because they said, well, they must be like sheep, and hamsters. They've got this melatonin and all the rest of it. And then actually people bothered to ask the Copper Inuit what was going on. And they have a very interesting societal structure. So during the winter months, they all huddle up and there's lots of communal living. But during the spring and summer, they leave, and young families leave and they disperse, and they have private time. And as they called it, they have the chance for intimacy. And so conceptions tend to occur during that June to September window, with births the following January to June. January to June, 39 births, compared to July and December, where there were only 9. So I guess what I'm saying is we don't think it's photoperiod melatonin. There could be other drivers, but we don't really know. And are we like hamsters? Well, certainly not. We have the capacity to generate an expanding and contracting melatonin rhythm, but we don't seem to use it to influence our reproductive status. It's not just annual rhythms. I'll just show you this quickly. So seasonality in suicide, peaking in April, May, June. Over 125 years, it's fairly consistent. Suicides in Japan, same sort of pattern, again, spring-ish. Then this is some work from Danny Rock, who's in Australia and has collected loads of studies from all over the world. Assaults on women tend to peak during the autumn time, and deliberate self-harm tends also to be associated with the autumn. So these are clear rhythms. They're not huge amplitude, but they exist. The drivers, we haven't any damn idea. We simply don't know. But in one case of human seasonality, we have a really clear signal, and that's seasonal affective disorder. So people who suffer from SAD, and it's a really crippling condition. It's really awful. And I've worked with the UK SADR Association, and you get depression, sadness, anxiety, increased sleep, just awful. And in fact, I remember a conversation saying, well, why don't more people with SAD commit suicide? And the person I was working with said, you don't understand. You are so fatigued. You are so lacking in motivation. You don't even have the energy to plan and then execute a suicide attempt. It's that flat. It's really awful. But it's correlated very, very tightly with day length. So this is some studies from the United States. And as you go from the long days of the winter solstice down in Florida of over 10.5 hours, and as you go north to 6 hours in North Alaska, less than 6 hours, 5 to 6 hours in Alaska, then you have a sharp rise in the percentage of the population showing full-blown SAD, which is different from the winter blues, which many of us get. But again, you see that beautiful climb. Same for the United Kingdom. So as you go north, you go from about 3.5 in midwinter in the south to northern Scotland to about 9% of the population. And again, similar statistics for the winter blues. So seasonal affective disorder and SAD seems to be associated with a shortening day length. And what's exciting is that there's an intervention. It can be treated with supplementary light using SAD lamps like this, of which there are many different sorts. This is one study that has been done. It's an early study, 98, where you see treatment weeks and then you see the placebo, and you see that in terms of remission, 20% with the placebo felt better. And this is classic in psychiatric illness, where you get a big placebo effect. And the placebo here was being told to sit in front of a positive ion generator. Then, if the light was in the evening, there was no statistical difference, or it barely statistically different. Perhaps by 4 weeks it was emerging. And then morning light had a significant impact upon the remission from feeling sad. And this is about 1.5 hours of light at 6,000 lux. And the duration and the amount of light change. So this is another study. This is not SAD, but it is in non-seasonal depression, but it shows the impact of light. And in this study, again, you get the placebo effect. And so you see a drop in changes of depression, -6 with the placebo. What's fascinating is that Prozac was only statistically significant from placebo after 8 weeks. And this is important to remember because it takes a long time for these antidepressives to kick in. This is the remarkable data. Light, which was 30 minutes of 10,000 lux first thing in the morning, was statistically different at 2 weeks from placebo and very different from Prozac at 8 weeks. The intriguing part of the experiment is that when they combined Prozac with light, the effect was additive. And that could either imply that light is having an augmenting effect on the same mechanisms or that Prozac has, or it could be using a different mechanism, and we simply don't know. And then this is another treatment whereby dim red light was compared to bright white light. And you see that after 5 to 6 weeks, you got a strong remission using light. So this is absolutely fascinating. How does light work? And despite what you'll read in the popular press, we really don't know. We know it's not melatonin, which is fascinating. So the original thought was, oh, well, SAD is caused because of the expanding melatonin pattern during the autumn. So people have removed that melatonin pattern and it's had no impact on SAD at all. So it doesn't, again, we're not using that melatonin pathway. What about circadian stabilization? Morning light is really important for setting the clock. And one argument is that it stabilizes, people with SAD just may have a sloppy circadian system and are less sensitive to light. And when there's less light around in the winter months, the circadian system seems to slide apart, and then you get a breakdown and increased levels of depression. And then finally, light acts as a serotonin selective reuptake inhibitor. That's actually turning out to be rather interesting in that SAD can, to some degree, can be alleviated with SSRIs, things like Prozac. And so the thought could be that light via unknown mechanisms is actually acting as a reuptake inhibitor. So it's preventing the synapses taking up serotonin. Serotonin is hanging around longer and therefore preventing the depressive effects. We don't know, but I think it's at least a model that can be analyzed. Okay, finally, I do apologize. That's really bad form. Biological rhythms encode environmental and physiological information to regulate and optimize multiple biological systems. That's the theme I wanted to develop today. And really, I wanted to sort of move away from the concept that most of us grew up with of the constancy of the internal environment, that the whole point of physiology is maintain a set point and constancy. No, biological systems are dynamic, very variable, and indeed the nature of this dynamic response to a varied environment, both physical and physiological, actually achieves physiological stability and physiological success. We talked about some specific examples in terms of the feedback loop of how circadian rhythms are generated and regulated by light. Then we talked about sleep regulation, some new and unpublished data on how that circadian oscillation, that circadian molecular rhythm, is being translated into an ultradian change in firing frequency that is underpinning the states of wake and sleep. We talked about seasonality, and where we are in at least animal studies is that we can have a pretty good understanding how day length, encoded by melatonin, can then be translated into a physiological signal that alters the pulse firing frequency of those GnRH neurons, and then the hormone release, which then changes reproductive status. And then we finish with humans. Yeah, we've probably got some rhythms going on, seasonal rhythms going on. They're of low amplitude these days, perhaps because we're just not outside enough. We're not experiencing natural photoperiod. Except for the fact that we've got one very clear rhythm, a seasonal rhythm that is dependent upon the amount of light we see, and that's SAD. Do we understand the mechanisms whereby light therapy improves SAD? No. And I think we're at a really exciting stage because we've now got some testable hypotheses. Thank you very much for your attention.